I'm very deeply honored to be here today. And I have to make these disclaimers required by NIH, and this disclaimer required by NIH. We're here today to honor the life and the scientific legacy of Athina. Her passing affected us all, struck us all. It affected me particularly because I had been very kindly invited by Mark and Athina to spend a couple of days with them this past summer at their vacation home in Greece. Alas, it was not to come. My assigned task by the conference organizers today is to review animal models of relapse drug-seeking behavior and to honor Athina's contribution to the search for anti-nicotine addiction medications, something that I have recently turned my own lab to also. I want to start with the reinstatement model of relapse to drug-seeking behavior. Many of you will be familiar with this model. Some, though, here in the audience, I know are not. It's based on the intravenous drug self-administration model, where an animal simply performs an operant task in order to get an intravenous bolus of whatever drug the system is loaded with. One takes the animal and allows the animal to self-administer drug until the animal reaches a criterion of response, stability, and then one deliberately extinguishes this self-administration behavior, this drug-taking behavior, by simply loading the system with saline or vehicle. Then, when the animal has reached a criterion of non-response, the animal is exposed to non-contingent drug or stress or environmental cues or contexts that were previously associated with drugging or drinking. And one finds that the animal goes back to profound drug-seeking behavior. It's not reinforced, it's not rewarded. Vehicle and saline are still in the system. Another animal model of relapse is the reactivation model. Some people are still calling it the reinstatement model, but I don't like that because it confuses it with the operant model. This is a model that's based upon condition-placed preference, where an animal receives drug, in this case nicotine, in one Cue-distinct chamber of a 2 or 3 compartment test device, and receives saline on the next day in the other compartment, and then after some repeated pairings, day after day after day, the animal is given free choice. If the animal goes back to the chamber where it received the drug, the inference is drawn that the animal is seeking the drug. If the animal avoids that, the inference is drawn that the animal is avoiding the drug, or the drug-paired environmental cues and contexts. One establishes a condition-placed preference, and then one deliberately extinguishes the condition-placed preference, and after the animal has reached a stable level of non-response, so that he is now neutral with respect to entry into either chamber, one again delivers the triggers to relapse, and one finds that the animal does, in fact, relapse to drug-seeking behavior. Yet another variant of cue-induced relapse is the so-called incubation of craving model. This, again, is based on intravenous drug self-administration with a very important difference. The animal is allowed to acquire, to a very stable level of response, the operant behavior of drug self-administration. And then instead of extinguishing the behavior, the animal is simply given a drug holiday, in this instance 2 weeks back in the home colony cage. And then the animal is reintroduced into the cues and contexts that were previously associated with drug-taking behavior, and one finds remarkable drug-seeking behavior, which interestingly incubates over time. And to any clinicians here, that will resonate, because that's what our drug addicts and our alcoholics who are in treatment tell us. The real problem in addiction is relapse. Mark Twain phrased it quite well. The 19th century American humorist, he said, "It's easy to stop smoking. I've done it hundreds of times." We've known since the 1930s and the pioneering work of Alcoholics Anonymous that there are 3 principal triggers to relapse. I've alluded to them already: drug, stress, and exposure to environmental cues and contexts. There has been a lot of work done to try and figure out what circuits in the brain are responsible for drug relapse. And what I'm going to give you is my admittedly idiosyncratic take on the literature, to which I and my lab have contributed in a very small way. I believe that the principal circuit in the brain that underlies drug-triggered relapse is this one highlighted, running from the ventral tegmental area up into the nucleus accumbens. I believe that there are 2, and this was alluded to by George Koob earlier. I believe that there are 2 principal circuits that mediate stress-triggered relapse to drug-taking behavior. One of them, and I agree with him, that while the locus coeruleus may be important, that the lateral tegmental noradrenergic cell groups are more important, and that they project forward into areas that we know are important in drug craving and drug relapse: the hypothalamus, the bed nucleus of the stria terminalis, the amygdala, and the nucleus accumbens. And there's a second circuit that runs from the amygdala down into the bed nucleus of the stria terminalis, uses CRF as its neurotransmitter. And of course, George has been extremely persistent in trying to find compounds that would effectively block or attenuate function in this circuit. And I believe that there are 2 important circuits, not to the exclusion of others, but that there are 2 important circuits that mediate cue and context mediated relapse to drug-taking behavior. One of them originates in the ventral hippocampus and projects ultimately to the nucleus accumbens. One originates in the amygdala and projects ultimately to the nucleus accumbens. I turn now to Athina's career-long quest for anti-addiction, anti-nicotine pharmacotherapies. Any cursory examination of her publications shows how incredibly important this was to her and how incredibly persistent and broad-framed her approach to this was. She studied metabotropic glutamate ligands of various types. She studied nicotinic acetylcholine receptor subunits. She studied NMDA compounds. She studied GABAergic compounds. She studied dopamine. She studied GABA B positive modulators. And the list goes on and on and on. This is a very small subset of her published work, trying to find compounds that would be effective for nicotine dependence, a very small subset. And I want to turn to some of her published work in this area. focusing on the GABA B receptor. This is a busy slide. I'll summarize it for you very quickly. Using the electrical brain stimulation reward paradigm, the ICSS model, a GABA B receptor agonist makes nicotine withdrawal more aversive, makes it more dysphoric, makes it more hypohedonic. But so does a GABA B receptor antagonist. And so does a GABA B receptor positive modulator. How do we reconcile this? I know that Athina was working on that, and it's tragic that her brilliance and her persistence could not be allowed to continue along this direction, because both GABA B receptor activation, I've just showed you this, and blockade exacerbated the anhedonic aspects of nicotine withdrawal in rats. Here's some more of her work. Repeated administration of the GABA B receptor positive modulator, BHF177, decreases nicotine self-administration while having little effect on food maintained responding, and significantly decreases cue-induced relapse to nicotine-seeking behavior while having no effect on cue-induced relapse to food-seeking behavior, using the reinstatement model of relapse. Those are simply some citations of confirmatory work. Are we here seeing a distinction between opponent process and proponent processes in the motivation to take nicotine? I leave that to people like George Koob, who would have, I think, very clear and profound and insightful comments to make. Unfortunately, Athina is now gone, and she cannot contribute to this ongoing work. And I thank you for your attention and your time.